of monitoring all clinical trial or basis of GCP. It's to manage the rights and well-being of human subjects and making sure that the rights and well-being are protected. And other purpose is that whatever trial data is reported is accurate, is complete, and verifiable from the source document. So source documents are the documents which are present at the site, and these are the documents where first information about the trial subject is noted, and these documents are used, and the data is matched with the CRF, and the data in CRF should be verifiable from these source documents which are present at the time. And it is to make sure that conduct of trial is in compliance with currently approved protocol or other documents which are approved by regulatory authority and ethics committee. So these are the primary purposes of monitoring. So now we are talking about monitoring. As Dr. Sunil rightly said, that with advances in technology, we are seeing many changes in the clinical trial, and monitoring is also touched upon by those changes. So the traditional monitoring is on-site monitoring, where CRA is close to the site and monitor the data at the site, spend 2 or 3 days at the site to completely monitor the data. But with the advances in technology, now we have remote monitoring as well as central monitoring. And I will talk about all these kind of monitoring in my next slide. So on-site monitoring or traditional monitoring. So traditionally, the pharmaceutical industry or CRO assess site's performance, going at the site and do an on-site monitoring. So the visits the sites, sit at the sites with the site team, with the PI, with study coordinator, review the source data and the CRS and matches them to make sure that everything is verifiable. And like I said, this approach is carried out by CI, and based on the monitoring plan or what is agreed with the sponsor, these visits can happen every month, every 4 to 6 weeks, whatever is agreed, and it also depends on the complexity of the study. And this approach relies heavily on the significant amount of source data verification. And there are several advantages of this type of monitoring, as well as there are disadvantages, which I'll talk about in this next. What are shortcomings of on-site monitoring? So there is a lack of real-time reporting. Patient comes to the site almost every day because every site 5 to 10 patients depending on what's their target, and they visit the site week or every month or every 2 months. And as I said, that the CRA visit the site every 4 to 6 weeks. So there is no real-time reporting of the data. CRA can see the data even he or she visits the site. The timelines of entry of data depends on study coordinator and site's workload. So it's often seen that entry of data in the CRF or in the source is not done on time. It's not done on the day when patient is visiting. So there's a gap between the patient visit and data entry. So this is one of the shortcomings. Then 100% source data verification, as per the latest research, is not very effective in analyzing the risk to the study, and it's very resource intensive. So CI has to visit the site, has to every corner of the country, every corner of the world. So a lot of resources are required in terms of manpower, in terms of finances. And this is a reactive approach, and ability to identify the issue quickly. So issue can be identified only by CRAs at the site, and so it takes 4 to 6 weeks to identify any potential issue. So because of which we are now seeing different kind of monitoring, which depends a lot more on technologies. And remote monitoring. As you can already understand by the term remote, that something which is done remotely. So in this kind of monitoring, the CRAs don't visit the site to review the data. Instead, the data monitoring is done virtually from the office itself with the use of internet, 5G host in India, and 𝐷𝗔𝗴𝗏. So we can use all these technologies and implement them in our industry as well. So this is being implemented very fast, and in last 2 to 3 years, the pace has increased because of COVID. And Dr. Sunil rightly said that COVID has impacted this industry a lot. And because of this, technology, the acceptance of technology in clinical trial industry has increased, and it is a type of monitoring they are doing. And right now, we are introducing clinical, that is traditional monitoring. Now, question. So, remote monitoring allows pharma. Question. If somebody wants to speak, can they mute their line? Okay. Can I please request everyone to mute the line if someone is not speaking? There is a lot of disturbance and distraction. Now moving to central monitoring, we talked about remote monitoring. So central monitoring is also a remote evaluation, of the data. It is performed in timely manner as per the agreement with the sponsor, and we have central monitors who are appropriately qualified and trained people to do this kind of monitoring. And this kind of review can be done using the analytical visualization such as graphs or with the help of dashboards to identify the trends or any anomalies or any outliers in the data which is collected at the site. In central monitoring, we don't have site for sending the source data, as a scan through an email or text. But we just review the data entered in the CRF through a dashboard and in a graphical format, which I'll share with you in the next slide. So this monitoring process provides additional monitoring capabilities. So doing central monitoring doesn't mean that for that particular study, CRA won't go to the site to review the data. So central monitoring and on-site monitoring are generally used 50% each in any of the studies. So a couple of data will be 100% SDR at the site. And there are a couple of data which can be reviewed internally, off-site, centrally. So what are the benefits of central monitoring dashboard? So as you can see here, let me take the pointer if I can. Yeah. So this is the CRA view of the data. So these are the CRA pages, and this is how CRA will view the data in the CRF. So if you can see, there is a separate page for vital signs, separate page for labs, separate page for EPRO or patient-related outcomes or things like that. So data is across multiple systems, and often the data is reviewed in silos. So when CRA is reviewing the vital sign, then his whole focus is on the vital sign, and he can correlate vital sign with the lab. But this is not the case with centralized monitoring view. So in the dashboard for central monitors, you will be visualizing data in terms of graphs and pie charts and things like that. So like you can see here, so if we are looking into a kind of score in a clinical study, so this is the way we will see that score. So mean patient score is in red line. The mean physician assessment score is in yellow line, and the delta between physician and the patient score is in green line. So you can compare all the lines, all the type of scores, And if there is any anomaly or any outlier, you can generate kind of catch it by viewing the graph, and you can with the CRA, the central monitor can alert the CRA that this for this particular visit for this particular patient, he or she believes that there is some error. And during the site visit, the next site visit, CRA will view this data is matching the source document and there is no error or fabrication of data. So use of centralized monitoring data. There are several uses, but the main uses are to identify the missing data, inconsistent data outliers, or variability or protocol deviations. Then to examine the data trends such as any variance, consistencies or inconsistencies or variability of the data within or across the site. So there are several sites in any particular study across the globe or across the country. So during the centralized review of the data, we can compare the monitor can compare the data coming out of several sites or several countries, and can look into if there is a variability of the data across the countries, and this generally helpful protocol is required. Then centralized monitoring can be used to evaluate errors in the data collection, to analyze the site characteristics and performance metrics. If there is a fabrication of the data at the site, or data is not entered on time, or data is not entered properly, this will give us the way the site is performing, and this will also help us to select the site for targeted monitoring. So if there is any particular site where a central monitor thinks, based on the data review, that immediate onset monitoring is required, then a targeted monitoring can be performed by a CMO at that particular site.
I request sir you to conclude in 2 minutes. Our next speaker is on board. Sir, please.
Absolutely. So risk-based monitoring, our main topic. So risk-based monitoring, or RBM, is an adaptive, like on-site monitoring. It's a mixture of both. So central monitor will review the data off-site in a centralized will go at the site and review the data, whichever has been highlighted with the central monitor, along the data which is initially agreed to be reviewed on site only. So the purpose of RBM is to use limited resources to where necessary, and generally we use 6R approach for RBM, which is risk identification, risk evaluation, risk control, risk communication, risk review, and risk reporting. We have two types of RBM. One is targeted RBM, which focuses on certain data points, certain sites, certain events that may have most risk, and argues that validating them will produce the best values, such as in the protocol, a screening visit where ICF has to be reviewed, or a baseline visit, or the randomization visit, or visit where primary endpoint has to be measured. These are the main visits, and these should be reviewed and SDV'd on site. The rest of the visit where there is no risk involved, or there's no measurement of primary endpoint, these can be reviewed in centralized manner. Then there is a triggered risk-based monitoring. This occurs when certain events, such as during the centralized review of the data, the central monitor finds out that there are several adverse events or deviation to a particular site. Then this triggers a detailed monitoring for that particular endpoints of this case. So targeted RBM and triggered RBM. So what is the impact of RBM on our industry? So it leads to reduction in efforts and expended on low value activities. So whatever are the low value activities, the industry is not putting a lot of effort to review them on site. getting more than what you invest. It leads to earlier detection of issues with more focus on resolution and prevention. Cost reduction, monitor or CRA will not grow that quick will decide the cost reduction or cost reduction of the project. Focus on study specific critical data. I'll give an example of primary endpoint. More collaborative cross-functional team approach. Benefits of RBM. So, like I discussed earlier, RBM benefits and preventing malpractice. This also benefits regulators to work closely. And sponsor companies enjoy more efficient allocation of resources. So you don't have to put all your CRAs in one study. With RBM, you can use CRA in study because you don't need that much time of CRA in a one particular study. And RBM also helps patients participating in a trial by shifting the focus to critical data and processes, right? Then technologies for Dr. Sunil also already talked about the DCT, decentralized clinical trials. So most of those technologies are used in RBM as well, such as electronic data capture or ECRF and the management system. And we have IoT interactive response technology, which we are using in the last several years in mobiles as well. Whenever you call a service center or a call center, that is an IoT. Then we are using central labs, quality management systems. We have electronic TMF, electronic diaries, EPRO, and nowadays we are using devices as well, such as one of us use smart watches. Those are devices, and then e-consent with these technologies such as electronic medical records are also used. So these technologies lead to integration. These are a couple of links for guidance of RBM. So guidance of RBM from FDA, from EMA, from MHRA, Clinical Trial Transformation Initiative. So these are the guidances if you want to go to the depth of RBM. And yes, if we have some time, I can take a couple of questions.
Thank you so much, Dr. Sumit Gupta. In a nutshell, you have told all the updates. Thank you so much, sir, for joining. Next, we have, thank you so much, sir. Next, on board, we have Dr. 𝗥𝗲𝗴𝗶𝘀𝗁, Quality Assurance Manager for Greater Melbourne, Victoria, Australia. I'd like to introduce Dr. Antu. Antu is having experience of more than 2 decades handling various portfolios such as QA, PV, project management with MNCs. He has diversified experience with US FDA, European Medicine Agency, 𝐃𝐮𝐿, NABL, and other national and international regulatory agencies. And she is trained CCP trainer, ISO certification trainer, and many more accolades to credit. So without taking much time, I would like to call 𝗥𝗲𝗴𝗶𝘀𝗁 to deliver. Thank you, ma'am.
Thank you. Thank you very much for this opportunity to talk to the students. It's a long time that I have least interactions after leaving India with Indian clinical research fraternity and the students, and I'm really delighted to have this conversation once again with the team. Without taking much time, I want to jump to the topic. Earlier, I thought that quality by design and critical to quality will be the topic for the conference, but later on, after knowing the target audience, I thought that corrective and preventive actions would be more useful. And today, I'm going to walk you through the steps involved in 𝐶𝐶𝐴 management. But before that, it's important to understand the topic. This topic has a great importance because your company's performance has a direct correlation with the 𝐶𝐴 management. If 𝐶𝐴 management has not been done effectively in any organization, one can see the trends in the share market. And there have been, couple of incidences, especially with Indian pharma market, where, corrective and preventive actions have not been taken appropriately, and because of which people have faced major regulatory hurdles. It could be, delisting of the company, or major regulatory complications have been witnessed by the companies. So corrective and preventive actions is an instrumental part. It's an integral part in any clinical trial, and it has a huge importance. And let's see what I have today. Happening.
You have to come out of full screen mode, ma'am.
So you mean I need to go back to the non-presentation mode?
Just come out of full screen mode, ma'am.
Okay, let me try. Let me give a try, please. Sorry for this. Okay.
Yeah, now it is moving, ma'am.
It's moving. Wonderful. So regarding the 𝐾𝐲 process, it's important for us to understand some background. So let's understand what is a deviation. Deviation is the departure from any good clinical practice, GVP, GMP, or regulatory requirements, and it could be departure from your study protocol or internal procedures. So whenever there is a deviation, there is a need for 𝐾𝐲, and the deviations could be of many types, and depending upon the grading, the actions are decided. So you have critical deviations, you have major deviations, you have minor deviations. So critical deviations are those deviations which has a direct impact on data integrity and patient safety. So if a deviation has impacted the two aspects, then you have to classify this deviation as critical, and the actions would depend upon the criticality. Secondly, the next definition is major deviation. Major deviations are those deviations which have the potential to adversely affect the data integrity or maybe the patient safety. So the deviation has not impacted, but it has the potentials, and this is the main difference between critical deviation and major deviation. And minor deviations are routine deviations which have no impact on data integrity or patient safety. So similarly, we have some definitions related to corrective and preventive actions, and corrective actions are those actions which are taken when the deviation has occurred. So the damage has already occurred and it is a reactive approach. So if if a deviation has happened at the site, you go ask them to record the deviation, do a correction in the data, or perform a procedure what is required as per the protocol or as per the study procedure, and you implement a corrective action. On the other hand, preventive actions are those actions which are implemented before the damage has occurred. So they are proactive in nature and they are more useful, and regulatory authorities have strongly suggested to implement preventive actions more effectively, so that the chances of recurrence of these deviations are minimized. So there is a need for 𝐾𝐲 management from regulatory point of view. There are various guidelines I'll show you in a while, which clearly explain the requirement, and so it is not a matter of choice. It is a requirement, and we are mandated to follow the requirements. From customer's point of view, it increases the customer satisfaction, and eventually you are giving comfort to your clients and customers that we have a robust system in place, and we can demonstrate that you can perform. From financial point of view is again very important. As I said earlier, all the deviations can fluctuate, the Sensex can fluctuate the dynamics of the company, because if your inspections are not handled well, they can lead to warning letters, they can lead to delisting and other stuff. So 𝐃𝐫 management has a great impact on the financials of the organization. From process point of view, the 𝐃𝐫 process stabilizes the 𝐃𝐫, stabilizes the process, and it's a good business practice. So these are some of the regulatory guidelines which clearly identify the need for 𝐃𝐫 implementation. You have 21 CFR Part 820, Part 211. Then there is ICH Q10, which identifies the need of a systemic implementation of 𝐃𝐫. On the other hand, ICH Q9 talks about the structured approach of implementing 𝐃𝐫. And it also says that the corrective and preventive actions should be implemented in such a way that it should be proportionate to the risk. If the risk is high, then you need to have more robust corrective and preventive actions. But if the risks are less, then you can take a moderate approach. So there are 6 steps which are involved, and I will walk you through each step which are involved in 𝐃𝐫 management. And before I end the presentation, I would also show you a practical case, but it will be very quick. Don't worry, I will not deviate from the time. So the first step in 𝐃𝐫 management is identification. The second one is evaluation, and then it goes to investigation and analysis. Then you implement an action plan, and then a follow-up is done. So let's see each and every aspect one by one. Regarding the implementation of 𝐃𝐫, it is important that the problem is defined, and the problems can be identified through various sources. The problem can come from customers as a complaint. It could be a process monitoring, like the previous speaker was talking about monitoring. So monitoring can also identify many issues or deviations. Then we also have a process of internal audits that can also identify issues or deviations. Your own staff member can observe deviations. Regulatory inspection is another way of identifying problems, and all those sources should have an evidence. So if you have a problem and if you have an evidence, then you can confidently say that there is a deviation, and then there is a need for 𝐃𝐫 implementation. So the evidence could be in the form of specific information. It could be a written complaint. It could be a trend. It could be in any form. It could be depicted through a data or document. And so while one should keep in mind that you're looking at the bigger picture, and if there is lack of ownership or accountability, then the 𝐃𝐫 identification can be greatly impacted. So the next step is evaluation, and we will cover this evaluation aspect in the next slide, which is investigation. So root cause analysis is the heart and soul of any 𝐃𝐫. So if the root cause analysis is done comprehensively, then you're mitigating a lot of risks, because with the help of root cause analysis, you will be able to identify the root, and based on the root of the problem, you will define your actions. You will also evaluate the impact of the problem. And so root cause identification is the sole purpose of 𝐃𝐫 management. And if root cause analysis is done correctly, then you're mitigating the risks. But on the other, is not done effectively, if it is not comprehensive, if it is not as per the requirements, then you will only deal with the symptoms. You will not deal with the real problem. So for effective implementation of any 𝐃𝐫 process, you really need to have a good root cause analysis. So there are various techniques that can be used for root cause analysis. It could be brainstorming with the stakeholders. It could be process mapping. It could be fishbone analysis. And the most simplest way of doing root cause analysis is 5Y. Our technique of 5Y is the simplest technique, and I will give you an example of 5Y in a short while. And these techniques have been found effectively because with the help of these techniques, you are able to see the issue in a bigger way, more comprehensively, more effectively. So these techniques are well known, and they have shown some good results. But again, every issue is different. But if 𝐾𝐲 root cause analysis is not done effectively, then it could lead to many issues. It could result in inconsistent investigation, or it could be not getting to the point. So root cause investigation again would be a very important aspect of any 𝐾𝐲 management. So the next step is action planning. So what do we do in action planning? In action planning, we make a very big comprehensive table. In this table, we identify the issues. It could be done in an Excel sheet or any tool you can use. And once the issues are identified, then you also need to identify the stakeholders who will be responsible for each and every action item. Then you define the timelines, you define the action, and then the accountable people will monitor this matrix closely on a regular basis. So once the action planning is done, then you need to communicate to people that these are the actions which have resulted from 𝐾𝐲 process, and this could result in change in the process, additional, or it could be enhancement of the infrastructure or technology. So stakeholder engagement is also a key aspect in 𝐾𝐲 management, and communication also plays an important role. So now you have a comprehensive matrix that talks about various steps, timelines, people who are responsible for each and every action. Now it is a time to implement. So based on the timeline, you implement those changes. You come in clinical research. If it is not documented, it is not done. So whatever actions you have decided, you need to document and demonstrate that the actions have been implemented. And like other your 𝐾𝐲 implementation will become a little bit tricky. You won't be able to track each and every aspect of 𝐾𝐲 management, because if there is one, it could be 10 action items. So there is a way to track each and every action or each and every issue. Then there could be lack of visibility. Otherwise, you will you will not be able to track your issues. Then the last and the important part of 𝐾𝐲 management is you have to ensure that whatever you have implemented and at a periodic stage implemented are effective or not. And this can be verified by checking the process. Secondary situation, which was never taken, is reviewing your 𝐾𝐲𝐴, which is very handy. If you want, I can share my slides later on for students, and they can read all those aspects at their own leisure. So let's go to 5Y technique. 5Y technique is a very simple technique, and this technique asks you to ask question why question 5 times, and then you will be able to find out the root cause. So this is a time-tested technique, and we will see an example here. So I really need a bigger screen because I won't be able to. This is my last slide, and I really want to put it in the presentation mode. Otherwise, I won't be able to see my stuff. So there is an example. I'm sure some of you are aware of protocols which are being implemented in clinical trial. So in this example, there is visit 2, and according to visit 2, some of the lab parameters have to be performed, and but it was seen that out of 7 patients, 3 patients did not have the correct lab exams. So when the monitor went to the site, he saw this discrepancy, and he asked the site coordinator, what is the reason for this mistake? So it was told that by mistake, these samples have not been taken, rather, sample of another visit was taken. So this looks very bizarre. This is not comforting to me, because, I really don't know whether you have implemented the protocol correctly or not. Then the monitor asked this question again. Why this has happened? Because then the coordinator gave an example, because the the site coordinator used something else instead of the right protocol, and the information was not correctly provided to her. Then the third time, the monitor asked the same question, and that time he realized that there is a transition in the team. The site coordinator who's doing these lab exams is not very well versed with the protocol, and there is a handover. Recently, the previous site coordinator has left the site, and the technician conveyed this information to the site coordinator, and then there is lack of training and lack of handover. So this is still not satisfying. Why there is lack of handover? When a new site coordinator is on board, then it is the responsibility of the principal investigator to train him or her so that he or she should be able to perform his task effectively. So at that time, it was found that the investigator did not perform the training effectively, and the handover process was very, very ineffective. So what could be the root cause of this problem? The main root cause of this problem is lack of investigator oversight. It is not lack of training. So lack of his involvement and his engagement. The investigator is responsible for oversight, and if there are transitions, he should be ensuring that the lab exams are done properly, the coordinator is trained effectively. So this is the end of the presentation, and I'm happy to receive any questions. Any questions?
We'll go to the quick Q&A session. If any questions are there, you can write in the chat box. As such, there is no questions. Thank you so much, ma'am, for your time and candor delivering this excellent talk on 𝐾𝐲𝐴 management. Thank you a lot.
Thanks. Have a nice sessions ahead. Bye-bye.
Thank you, ma'am. Thank you so much. Thank you. Next, I would like to call our next speaker, Dr. 𝐵𝗫𝐻𝐾𝐲𝐴. 𝐵𝗫𝐻𝐾𝐲𝐴 is serving as CEO with the NIMS Rs and CEO leading trainer to 𝗥𝗲𝗴𝐻𝗔𝐺𝐴M and triple 𝐿SSDC and has over 23 years of contract research organization experience in regulated bioanalysis or preclinical and clinical development, including bioanalytical and biochemistry studies. He has trained more than 200 plus GCP inspector of various regulatory agencies, international famed China, Thailand, Malaysia, Indonesia, Singapore, Philippines, Qatar, through WHO CQT workshops and over 1,000 clinical research professionals, over 500 bioanalytical, 400 clinical, 100 quality professionals of CRO, pharma, R&D, and international regulatory agencies. With this short introduction of Dr. Manish, I would like to call you, sir, to start the talk.
Thank you so much, Dr. Kose, for nice introduction. And I'm really privileged and honored to be a part of this conference. And thank you for inviting me to talk on maybe a very strong and important topic, which is related to quality in clinical research. So with this, I'd like to have maybe share screen rights. If it is there, it is good. So I would like to share my presentation. Okay.
It's 𝗥𝗲𝗴𝗶𝘀𝗁𝗁.
Are you able to see my screen now in an appropriate way? So again, let's come to directly to the topic. I born and brought up in CRO industry, as already introduced by Dr. Kose, but then spent most of my time, almost all time, in regulated environment. So whatever I've done in commercial research, it is in regulated environment for either for US or Europe. Sitting here in India, so never been work for an MNC or outside company, but we have done all the good work from here. We published more than 50 research articles. So let's begin the topic. Little bit, okay. So it's a standard slide. I'll not spend much time. This is just to connect and get your focus into drug discovery process. And where you can see it is taking close to 15 years time, 10 to 15 years. Now this is being shrunken to maybe I say 8 to 9 years time. Earlier it was around close to 20 years. So with all the advancements, which always I feel as a researcher, it can be a pain for somebody who is not passionate about research, because there are a lot of changes, a lot of updates keep coming in research field. And research has become especially, okay. If my video is not visible, then I think it is a challenge to. So am I visible, doctor?
Yes, sir, you are visible as well as audible, both.
Okay, because somebody gave me message, my video is not visible.
No, sir, you are very visible and
okay, fine, sorry. So I was talking about the drug discovery process, maybe a research. If you want to discover a drug, it takes somewhere around now in current scenario 8 to 9 years, and it is all because of advancements. And when I talk about advancements, it directly related to the dynamism. And when I talk about dynamism, it takes a lot of dynamic efforts, dynamism of a researcher. So what does it mean? I keep communicating this message and keep trying to keep telling you what does it exactly means. Coming back to this slide, we have very important steps to discover a drug, from simple chemical to declare it as a drug, effective drug. Right? So drug discovery, like chemical lead identification and then preclinical experimentation to be done for 5 to 6 years time, then maybe 6 to 7 years time for clinical trials. Then it's under review of regulatory agencies, basically what we target the market and regulatory agencies based on that. Finally, when everything is in place, process is in place, approvals are in place, we go for manufacturing. And there also we have a lot of regulations, right? Like GMP. So now here, I would like to focus on clinical trials, clinical research basically, and the clinical, what we say in short, clinicals. When you talk about clinical trials, I'll certainly move on to the quality aspects of clinical trials, but then let's understand we all know, but still I'll just try to remind, we have different kind of studies like PK studies, pharmacodynamic studies, and in clinical research, I have seen people who are not able to talk much about the bioanalysis. When you talk about PK in healthy or patient volunteers or basically patients, healthy volunteers or patients, and if it is a PK study, PK endpoint, then we need to have bioanalysis also in place. So regulated bioanalysis is also an integrated part of clinical research, what I believe. So when you talk about regulations, it's a general belief, like we don't need much regulations and research processes. When we have research, typical basic research, people have that mindset. But believe me, with the passage of time, with my experience, I realized you need more regulatory controls, quality controls, basically, when you are doing pure research. Nobody is watching you, nobody is asking you for the results, but still you have given time, and then you are doing a lot of experimentation, and based on that, you are trying to say this is our lead compound. It is not only related to chemical, I'm saying, even for clinical aspects, or when you have lab experiments, you have a lot of good time for development, what we are spending. And sometimes there are no controls. Nobody is asking how much time you spent. But that time, if you don't have quality controls or quality aspects in place, maybe systems in place, actually you are losing sometimes a very good molecule or leads, and you have to throw them out because you cannot repeat those experimentation. You cannot get that results, the reproducible results, until I have that reproducible presentation or performances. You cannot say this is going to be our lead compound. I don't know how much I'm able to communicate, but trust me, I believe you need quality controls everywhere in every steps of drug discovery process. But to shrink this point to the presentation, I would like to say everything starts conventionally, everything starts from preclinical, where we say we have very good controls on experimentation so that we can reproduce the data, and we can show the internationally accepted data across, right? Then when we move to the human research, we have another stronger compliant practices, which are called GCP, and then GLP, obviously. GCLP is something which we are going to talk about, which are probably more suitable to the clinical research. Standard process, trial, any standard trial, you know, we have protocols, as Antu also clearly mentioned. We have different documents, what we need to conduct the trial, like protocols. But then if you talk about hardcore operations, we have various operations like registration, screening, pre-check-in, check-in, study, pre-check-out. Pre-study process, it's a part of study. Conduct, basically pre-study process, then dosing we have to do in study, and there only we have a lot of monitoring aspects which are to be taken care through monitoring, a lot of study aspects. Sample collection, plasma separations, and then finally storage. And if it is a PK endpoint, then bioanalysis comes into picture. Clinicals, basically, I believe, consists of various aspects like clinical operations. There we have trial design, standardization, which is a very important point from quality control front. PK samples like trial sample analysis, and quality practices like GXP. So X can be GC documentation, or like manufacturing, so many things can be X. So we say basically. But if you talk about GCP basically safety, rights, and well-being of subjects. About regulations, based on experience, I say most of the pharma companies originated from India. They are targeting European market, which is EMA, then US FDA, and then our local market like Indian market, DCGI, and so many other countries, but I have taken the principal example. Now, when we talk about bioanalysis, we have various processes, but importantly, method development, method validation, and subject sample analysis. Subject sample analysis starts from the collection of samples in clinical facility. From there, the samples will be transferred to the lab. There we store the samples, and then finally, when somebody wants to analyze those samples, as they have to retrieve the samples, they have to thaw the samples on room temperature, they have to prepare 45 standards like calibration curve and quality control samples, sample batch processing, then they have to do analytical batch loading run on machine, analytical batch acceptance criteria. So we have quality management system, various SOPs, policies, work instructions to control all these steps, okay? And to reproduce these all these steps and the results, we have to have quality management system in place. But importantly, as I mentioned, method development is another area. There sometimes people follow their own, processes and policies. There also we need very good controls. But basically, when we talk about development, we develop method for sample prep, we develop method for chromatography and LC-MS in today's as per today's requirement. These are the steps, and based on these steps, you can develop your controls. Now, clinical research, I'm a little bit diverting towards regulated bioanalysis, because if I want to explain you dynamism, then bioanalysis part is something which is more relevant because we have a lot of advancements. I just compared GCP and GLP, especially for regulated bioanalysis. GCP, you are not able to see even 10% difference what we were following 2 decades before. So it's a 2 decades time. Like I started my career in 1998, and from there to today. But if you say the GLP and regulated bioanalysis part, because of this technological advancements, machine, what we are using, equipments, their advancements, and overall regulatory aspects, they also learn from basic advancements in research and equipments, and even the learning of the people. So regulatory bodies are also coming with the advancements. So that is probably adding these kind of happenings, adding a lot of dynamism in not only to bioanalysis, because bioanalysis again is a part of drug discovery process and the clinical research, basically adding a lot of dynamism to clinical research. But to explain you, I'll give you a little bit glimpse what kind of dynamism you are expecting even in future from in regulated bioanalysis. So basically, first dynamism you can expect from the scientific front, because a lot of technological developments are happening. Now people are talking about pediatric doses in clinical research. People are talking about less side effects, means minimum dose which can be effective. So when you have minimized the dose, you need more sensitive techniques to detect the quantitation or to do the quantitation of that drug in biological matrices, which are coming with those samples are coming from clinical trials, right? So basic science, because of this, what we are having vital component of reg bio. So basic science, you understand there are going to be a lot of changes and positive changes and advancements. So it is adding dynamism to this area. Quality, GXP, we talked about scientific advancements, means when there is an advancement, there will be publication, there will be conference like this, what we are having today. We have white paper. Based on white paper experience and collective experience, even regulatory bodies can come and talk to us, and they can say, we are getting some of the knowledge from this conference and improving our regulations. So basic science, quality practices, advancements may compel regulatory body to adopt new practices. So regulations are also getting changed. But all said and done, while following everything, good science, good practices, and good regulations, ethics is going to be the focal point from all fronts when you talk about clinical research. Scientific advancement, as I mentioned, better scientific clarity. It is coming through a lot of scientific advancements, publications, okay. Extended experimentation for bioanalysis, again, due to quality practices and industry practices. Industry accept the practices first, then sometimes even regulatory bodies comes and they say, okay, we are going to implement and we need this kind of experimentation now. Understanding of allied sciences, not only a person who is working for a bioanalytical lab, he is not only focused on bioanalytical, he should know the clinical, what is going on there, and the formulation science. If he or she understands that, it is going to be helpful to ask 5 whys. So when you go investigate something, as Antu also mentioned, we can ask the right questions on right time to the right people, right? So that is allied science, I mean to say. Regulatory advancements, when you talk about earlier, we used to learn and having only 483s warning letters. Then when we have warning letters, these leads to transformation in regulations also. Sometimes based on findings, regulatory findings, regulations, regulatory body comes and they say, we are going to change. We are now expecting some more from you as a client, as executors. Then they can issue new guidances. Like EMA came in for bioanalytical method validation. EMA came in 2011, and with the first guidance was 2001 from US FDA. Then EMA 211, 10 years after, and then NVISA 2012. US FDA came up with new guidance for bioanalytical method validation 2018, and now recently ICH guidance M10, ICH M10 for VMB bioanalytical method validation released in 2020. And to surprise to all, not at all a surprise, because I work for this getting common harmonized guidance for different global harmonization teams. So I know the process. 10 years back we started, we get the results. Now it's a common, and this the EMA has already superseded that guidance with this M10, and now US FDA also published in their registrar. So it means M10, ICH M10 is going to be uniform guideline to validate any method and to submit the data. Everybody has to accept this. So that is a very good progress we made. Good quality practices, as I mentioned, GXP and X can be C, L, M, and D. D means documentation. So when you talk about these kind of practices, what kind of standards we need? Now, before I go to that, scientific advancements, a lot of advancements in extended selectivity testing happened, like matrix effect, stability, cross-contamination, biotransformations, which is again molecule and interaction-based, body molecule interaction-based kind of phenomena. So when this is happening, a lot of new things are coming into picture, like back conversion, metabolite, drug to metabolite, metabolite to drug again. But anyways, so those are the part and driving factors, and adding dynamism to research, technological or technical advancements from scientific front. Regulatory front, I divided regulatory era into 3: pre-regulation era, post-regulation, and dynamic. And this is basically based on bioanalysis. So pre-regulation era, there we didn't have anything except of nothing. So some white papers conferences happened, white papers came. Then post-regulation era means there we got first US FDA guidance. And based on that, we run our show for 10 years. And then after almost 10 years, we got NVISA and EMA guidances. And today we have ICH M10 harmonized guidance. So even regulatory front, if you see, especially bioanalysis, we have a big change. And this is one slide which you can see later on. Even for one experiment, how they are changing. It's a drastic change in 2001. And then if you see 2022, what they are talking about, how much they are demanding for single experiment, which was not even known when we started our career. Progress, again, this is to understand dynamism. We need good, once we understand this is a highly dynamic area like clinical research, we need to have proper quality management system. These are the few characteristics. I'll not read this slide for you, but sound scientific, the base for developing quality management system, we have to have sound scientific base and probably best clinical and bioethical quality practices. Means sometimes regulatory bodies will not ask you much. You have to have your own experience, attentiveness to discuss among your groups and friends circles, which are good. From there, you can get good practice, which is really may become industry practice, and then later on even regulatory agree and start asking on that. So this is the way we have seen in our career. So if we are attentive, we can develop our system, which are probably way ahead what even regulatory or quality today is they are expecting. This system has to be rigid enough to have consistent results to give, and flexible enough to cope up dynamism of not only bioanalysis in the clinical research. So this should be fit for purpose. Now, why I'm talking about dynamism and change and advancement, you understand the necessity is the mother of invention, right? So when I say quality practices, I don't see for bioanalytical lab any practice is really standard and good, because when you talk about GCP talks about compliance, okay, and basically the rights, safety, and well-being of trial subjects. When you talk about GLP is a managerial concept and talks about organizational structure and conditions, study conditions, right? But where we get the idea how we handle the clinical trial samples, there is a gap. Alone, if you talk about alone, GCP or GLP or GMP can handle the pressures of clinical research or bioanalysis. I say no. So we need a quality standard, which is hybrid, maybe new. By GCP, you know, they are talking about well-being, other systems. GLP talks about laboratory organization and other things. GMP talks about investigations. GMP is best if you want to learn how to investigate the challenge. Bioanalysis involves not only one thing. Trials involves human samples, analytical equipments, laboratory, equipment qualification. Yes, it is because the equipments are there, we need equipment qualification. Failure investigation, when you are conducting something, there are chances of failures also. So failure investigations, root cause analysis, impact analysis, kappa, everything. So this comes better understanding you can get from GMP. Analytical equipments laboratory you can get from GLP, and human samples, how to handle human and human samples, GCP talks about. So standalone is not going to be useful. Quality practices, when you talk about, we need an hybrid standard, which is good clinical laboratory practices. Fortunately, unfortunately, this came in 2009, 2003 first, and then later on 2009, WHO GCLP. Now we have three other from MHRA and others, 3 GCLP guidelines. Still in India and developing countries like us, very few people are really able to implement this system, this practice, which is actually more suitable. Because this will ensure the reliability, this is ensuring reliability, quality, integrity of work and results, GLP expectations, and
sorry to interrupt you, sir. Our offline guests have been arrived, so I request you to conclude in 3 minutes. Sir. Please, sir.
Oh, okay. Dr. Kose, you are messaging me something?
Yes, sir.
Our guest has arrived, sir. Offline, we have to start the function. So, sir, please conclude, sir. We have 1 minute.
So give me 5 minutes. I'm going to conclude this.
Sir, 1 minute, sir, because they have arrived in the auditorium.
Okay, fine. So I'm almost done. Okay. So I was talking about this is a hybrid practice. Now, my topic starts with quality. So I talked about advancements, dynamism, and dynamism. If you want to understand then advancements, you need to understand advancement, scientific advancement, regulatory, and quality practices. I try to give you that idea. And now, why I was talking about that advancement, because quality, what we believe, is not stagnant kind of thing. It is a moving object. And when you talk about something is moving, what kind of attitude and acumen you should have. So you have to have a continuous improvement kind of acumen. I'm escaping the QMS part, but that's fine because I believe we have already covered this. But maybe one liner to when we develop the quality or QMS, you need to understand what you are saying, what you are doing, you should say, and whatever you are doing, you should write, prove it and improve it. So continuously, that is what the basis of even implementing the QMS. The QMS has to be implemented so that you can get repeat results consistently with the accuracy. And that is what the core idea of having QMS. When I talk about dynamism, I talk about quality in clinical research. I say quality is something which can be defined with the best way you say it's a moving object. So as clinical research is highly dynamic entity, I believe, hence progressive mindset and continuous development are certain things which should be there in clinical researchers' mind. And those are the tools which will probably able to align you with the dynamism of clinical research, because it's a highly dynamic area, highly advanced area. A lot of advancements are coming, so we have to be attentive throughout. So with this, I would like to conclude, and thanks for your time and patience listening me. And I don't know whether we have time to have some questions, or I can take offline also. Thank you so much.
Thank you so much, sir. A brilliant session on quality management, sir, you have delivered, and definitely it is a YouTube live. So, sir, if any Q&A session, it comes to us in the chat box of YouTube, I will be sharing it to you, sir. And thank you so much, sir, for taking really good content to all of us online. Meanwhile, the time constraint was there, so next time we'll take it on the notes. And thank you for time and candor, sir.
Thanks.
You so much.
Take care.
Once again. Thank you so much. Thanks all the audience, virtual audience over here for joining. And now it's time for offline session at the auditorium. So we'll be meeting over there. Thanks all of you for joining this session. And I would like also thank Dr. 𝗥𝗲𝗶𝘀𝗁, sir, especially chairing this session for joining this session. Thank you so much, sir. You may leave now. Thank you so much, sir. Thank you.